And thanks everyone for being here today. It's great to see such a wonderful turnout. It's my tremendous pleasure to introduce Danny to all of you. Danny joined the biology department at Western in 2012. This was her first faculty position. Before that, she finished her PhD at the University of Toronto in 2008, and then spent a few years as a postdoc and a research associate at Duke University before we were able to nab her. Danny's research looks at the physiology of plants, mostly trees, within the context of ecology and global change. So her research is really special in kind of spanning and integrating across all levels of biological organization, from biochemistry right up to global ecology. And her research program has also been tremendously successful. So Danny has over 70 peer-reviewed publications that have been cited, I think a total of more than 5,000 times. She's received over $4 million in research funding through her career. She's been an editor on the boards of several journals, including Global Change Biology, which is one of the highest impact journals in ecology and environment. And this is all someone who, hasn't even been in her first faculty position for 10 years. So I think it's really quite astounding. And so not surprisingly, Danny has received many awards and accolades already in her career, too many for me to mention all of them. But notably last year, she was included in Clara Veit's list of highly cited researchers. She is a member of the Royal Society's College of New Scholars, Artists, and Scientists. I think I got that right. She's been awarded an early career award from the Canadian Society of Plant Biologists. But interestingly, when I asked Danny kind of what were the awards or accomplishments that she was most proud of, the prize that she mentioned was actually an in-house award from the Society of Biology graduate students for the most supportive faculty member. And I think the fact that this was the award that meant the most to Danny really tells you a lot about her as a person. And I think explains why she's such a great collaborator and a great colleague. As successful as she is, Danny really supports the success of others very strongly as well. So it's my tremendous honor to be a colleague of Danny's. Congratulations, Danny, on the Science Prize. And I think I speak for all of us when I say we're really looking forward to your lecture. That's all I have to say.
All right, I think that means it's probably my turn. So I don't think I could possibly actually live up to that introduction. So thank you, Dusha. That was really kind of you. So I'm going to share my screen and see if we can get this working properly. So now let me see if I can see everybody. Can somebody give me a thumbs up if you guys can see the screen okay? And it looks like it's just the one screen. Thumbs up. Okay, good, awesome. Yeah, it's really an honor to receive this award, and I'm really looking forward to sharing a little bit more about the sort of work that we do in our lab with you today. So I realize it's a broad audience. I've tried to capture enough background so you can understand the material, and I also am sort of now ambivalent about the title that I chose, because I chose this back in December. But hopefully this is the last time you have to see the word Trump on any sort of a slide. So warming trumps CO2 and looking at climate change impacts on plants. Going here. So atmospheric CO2 concentrations have risen by about 50% since the Industrial Revolution. And we started monitoring atmospheric CO2 concentrations directly back in the late 1950s. And this is a figure that goes up to about September of last year, showing CO2 concentrations on the y-axis, and then you can see years across here. So there's this steady increase in CO2 concentrations. And one of the things I find interesting about this is that the red dots, the red line, is actually the week-to-week measurements of CO2 out in Hawaii. And that fluctuation that you're seeing, as opposed to the smooth line, which is the black line, is showing really the effect of vegetation on atmospheric CO2. So every summer, the forests, especially in the Northern Hemisphere, they're so vast that when their leaves come out and they become active and they turn on photosynthesis, they absorb CO2 to the extent that globally, CO2 concentrations actually decline. And then every autumn when the leaves shut down their metabolism or senesce, then that goes back up as CO2 fluxes out from the leaf in respiration overwhelm that signal. Now, of course, CO2 is a greenhouse gas, and temperatures have actually already increased globally by about 1 degree Celsius since around 1900. And if we continue on this sort of a trajectory, then this is the sort of future that we're expecting by the year 2100. And this is a map showing annual mean surface air temperature changes. This has been put together by the Intergovernmental Panel on Climate Change, the IPCC. And any areas that show blue mean that you're expecting a cooling trend, whereas of course these yellow to purple indicate that you're going to get warming. And so first of all, the implication is that of course there's no blue. And secondly, you're expecting around a 3 to 4 degrees Celsius increase in mean global temperatures, but that's much greater at high latitudes. So you can see that areas in Canada here, we're expecting temperatures much more around +7 to +9 at our high latitudes, and even up to +12 in the Arctic. So these are enormous changes. And of course, I'm really interested in the interaction between climate and plants, and that's for two reasons. And the first is because climate is a really important control on where you find certain species and how productive they are. So this is just a map showing for a single species, so for aspen, this is where you find it approximately now. So the dark green shows the core of its distribution, and then the lighter color shows sort of the extremes of where you find that species. And if you assume that temperature and precipitation are the primary controls on this distribution, then the expectation is that within 50 to 80 years, you're expecting to see a complete movement of that aspen forest up into what is now tundra. So you can see this dark green strip is really quite high up. And basically where you currently find that forest, there simply won't be any aspen left anymore at all. 50 years is a pretty short time for this to happen, okay? And looking at it from the other direction, thinking about what our climate might look like, what our forests might look like, if you look at distributions in the recent past of forest types, and this comes from the US, so of course Canada just doesn't exist in the map, but you can imagine where London is here. These different colors indicate different types of forests. And the one I'd like to draw your attention to is the red, and that's your maple beech birch forests. That's the type of forest that we basically get in this region. And again, if you predict forward by the end of the century, then that forest type disappears. And instead, what we end up with is a forest and a climate that starts to look like Virginia and North Carolina. And that's what we're expecting again in a pretty short period. So the second reason to be interested in how plants and climate interact is because plants also affect climate. So I sort of indicated that when I talked about the oscillations in CO2. But if you look at these big predictions that are made in these models by the IPCC about future climates, you need to account for socioeconomic trajectories of what we do, how we decide to move forward with our energy systems and our population. But you also need to account for really large carbon fluxes that occur between plants and the atmosphere. And those fluxes are dictated primarily by two physiological processes, which are photosynthesis and respiration. So those fluxes are really big. They're some of the biggest fluxes in the global carbon cycle. And for terrestrial plants, photosynthesis absorbs 123 billion tons of carbon every year out of the atmosphere. About half of that every year is then released from plants through what we refer to as autotrophic respiration, so respiration from plants. And another 60 billion tons of carbon is then released out of soil microbial processes mainly, so respiration from microbes and animals. And so to put those sort of fluxes in context, anthropogenic CO2 emissions from fossil fuel burning and also from land use change are approximately 10 billion tons of carbon a year right now. And so really the scale that I'm talking about here is that if you have fairly small changes in photosynthesis at this global scale, they can easily be on the same sort of value scale as the anthropogenic emissions that we're doing right now. So in other words, if climate change suppresses photosynthesis by a relatively small percentage, that's going to be an enormous change in the global carbon cycle that's going to affect climate possibly on the same scale as the emissions we're making. And just to sort of drive that home, this is a figure we published fairly recently. These are the exact same numbers I just showed you, where you have 123 billion tons of carbon being absorbed by plants and 60 gigatons of carbon coming back out. another 60 gigatons of carbon coming out from soil microbial processes, and then humans emitting about 10 gigatons of carbon. If we just look at those processes and ignore all of the other things in the global carbon cycle, the balance of those fluxes is around 7 billion tons of carbon every year accumulating in the atmosphere. And this is where that rise in CO2 comes from. But if that rise in CO2 and the warming that's associated with it ends up suppressing photosynthetic rates by relatively small amounts, even if that suppression of metabolism also suppresses respiration by a small amount, and if nothing else changes, the difference in the amount of CO2 that accumulates in the atmosphere ends up being, like in this sort of thought experiment, a 23% increase in the rate of climate change. So relatively small changes in these processes can scale to have enormous consequences for the planet. And that sort of leads to the question of, well, how well are we able to represent these processes in these large models? And a couple of years ago, I was part of a working group that pulled together photosynthetic physiologists with these Earth system modelers who pulled together these large scale models. And we said to them, if we give you just a temperate tree and we say to you, we'd like you to, at current CO2, here's some index of leaf nitrogen, so an index of the metabolic rate of the plant. How does photosynthesis in your model respond to short-term changes in temperature, just from minute to minute? And this is what we got. So as they ran these through the different models, here you have assimilation of CO2, so photosynthesis, versus just instantaneous changes in leaf temperature over minutes. And each of these lines represents a different Earth system model that is incorporated into those sorts of figures that I showed you from the IPCC. And so you can see that in some of them, photosynthesis peaks at around 15 degrees Celsius. In others, it just keeps continuing even up at 40 degrees Celsius. And even when you get a sort of moderate peak here at around 25 degrees Celsius, which is realistic, there's a lot of difference between the actual rates of CO2 that those models are assuming. So in other words, there's a lot of scope for improving how we are able to model and estimate what future photosynthesis is going to look like. So what I'm going to talk to you about today are two things, and then we're going to combine them. So the first is, how does rising CO2 affect plant carbon uptake and growth? And then we're going to talk a little bit about how does rising temperature affect plant carbon uptake and growth. And then a lot of my recent work has really focused on how does the combination of rising CO2 and temperature affect plant carbon uptake and growth. So it's much easier to control simply one variable than it is to look at the combination of them. But of course, in a future climate, plants aren't going to see just rising CO2 or just rising temperatures. Instead, these happen together in concert. Okay, so let's start with the CO2. And just to make sure we're all on the same page, I'm going to give a really very simple explanation of what photosynthesis is, because I realize not everyone here is a biologist. And so if you take a leaf and then you take a cross section of it, for the purpose of this talk, we're really only interested in the CO2 fluxes. And so what that means is that CO2 has to get from the outside of the leaf in, and it does that through these little pores called stomata. So you have to supply CO2 into the leaf. Once it's inside the leaf, that CO2 has to be fixed by the biochemistry of photosynthesis, so by the enzymes involved in photosynthetic processes. And so we're just going to keep it that very simple definition of sort of supply and demand, really, for CO2 inside a leaf. And when you think about elevated CO2 then, if you think about the machinery of photosynthesis, the way that that operates is that there's a key CO2 fixing enzyme known as rubisco. And the basic reaction is that rubisco fixes CO2, which is here, to its substrate, REBP. It then uses energy and reducing power to turn that into carbohydrates that are used to fuel the metabolism and growth of the plant. So again, at a really simple level. What that means is that if you provide more CO2, you provide more substrate to this reaction, which means that the reaction rate increases, and so you stimulate photosynthetic rates. So under high CO2, we get higher photosynthetic rates, and therefore, you should see more carbohydrates accumulating, and you should also get faster plant growth. And when it comes to that supply side of the equation, I talked about stomata, and this is a close-up of what they look like on the leaf surface. So these are these little pores on the leaf surface that allow CO2 to diffuse into the leaf. And they're also responsible for water diffusing out of the leaf. So under high CO2, what happens is the stomatal aperture closes to some extent. It's not quite open as widely. And that means that you reduce the evaporation of water from the leaf surfaces at high CO2. And so the expectations from these sort of first principle knowledge of plant physiology is that under high CO2, we expect plants to have greater CO2 uptake, more photosynthesis. We expect less water loss, and we expect to see higher growth rates. So all of those seem to be pretty good things. But what happens is you end up with acclimation. And so this is a complicating factor that when you grow a plant at high CO2, it alters its biochemistry and its physiology. So for terrestrial plants, nitrogen is often a limiting nutrient. And this is why you apply fertilizer to your house plants or out in your field. And specifically that enzyme rubisco is approximately 30% of leaf nitrogen in many species. It is the most abundant enzyme in the world, and it's usually the most abundant enzyme inside a leaf. So it's a huge cost for nitrogen. And if that enzyme is operating at high CO2, where there's a lot of substrate availability, the plant can actually downregulate the amount of rubisco it makes, which saves a lot of nitrogen that it can then reallocate to other physiological processes. So you can think of this as sort of an optimization issue. And this is indeed what we see in plants that are grown at high CO2. You see lower leaf nitrogen concentrations, lower rubisco concentrations, and therefore that stimulation of photosynthesis we expect to see is actually suppressed in plants grown at high CO2. So it's basically like the idea that if I feed my 4-year-old only candy and only sugar, she's getting lots of carbohydrates, but she's not getting protein and all of the other components she needs to build a healthy body. And plants are the same. They need carbohydrates, but they also need nitrogen and phosphorus and other components to build their tissues. So giving them more carbon can be helpful, but it's not the only thing they need. So I'm just going to show you a little bit about how that plays out at a large scale. So this is an experiment that I worked in during my postdoc. So this is an aerial view of the Duke FACE experiment. And FACE stands for free air CO2 enrichment. So you have 6 experimental plots. And this is a large pine forest here. And in 3 of these plots, the ring, which is a ring of pipes around this intact forest, is blowing pure CO2 into the plots, depending on wind speed and wind direction. And in the other 3 plots, it's blowing in ambient CO2. So 3 of these plots are experiencing high CO2 out in nature. They're in natural soils, they get natural precipitation and herbivory. And the other 3 plots are ambient CO2 controls. And just to show you what that looks like from the side, this is a view of one of these rings where you can see sort of the structures holding up the pipes. And then the pipes have holes in them that blow CO2 out into the system. So I mentioned that high CO2, you do expect to see an increase in growth. And this is indeed what we see at the experiment over those first few years. So the productivity above ground of the forest, this ANPP, was stimulated by about 30% on average across the first few years of the experiment. So here you have that productivity measure. Here we have years. And the white dots show the ambient CO2 plots, and the elevated CO2 plots are here in black. So before the CO2 is turned on, the plots have similar productivity rates, and then you turn on high CO2 and basically tree growth is accelerated. But this is a relatively fertile soil where there's a lot of available nitrogen. And so what happens in other experiments, and this is another FACE experiment that was at Oak Ridge National Lab down in Tennessee. Here again, you see that net primary productivity across the years of the experiment, ambient CO2 plots in white, elevated CO2 plots in green, and the numbers show the percent stimulation in productivity due to elevated CO2. So in those first few years of the experiment, you get a big stimulation from CO2, but over time, acclimation kicks in as the plants readjust their physiology and their stoichiometry, and you end up with plants that are basically producing the same amount of wood every year and the same amount of foliage. So that treatment isn't nearly as strong when it occurs over time and there's acclimation. Okay, so let's talk a little bit about how rising temperatures affect plant carbon and growth, and then we're going to combine them at the end here. So just like most biological processes, photosynthesis is temperature dependent. And this is a figure from a synthesis that I did with some colleagues a few years ago, where we gathered data from over 100 species and characterized what are the average temperature response curves of photosynthesis. So there are three types of photosynthetic pathways in plants. C3 is the predominant one. The vast majority of our plants are C3. So we're just going to focus there. And that's this green line. So as you increase the leaf temperature, you can see that originally you get this stimulation of photosynthesis up to a thermal optimum. And that's because you get increased enzyme efficiency with increasing temperatures. And then you start to get this decline as you get to these higher temperatures. And the thing I want to point out is that this isn't a linear effect, right? So unlike with high CO2, the direct effect of warming on carbon uptake in plants depends on where you start. So if you're a C3 plant that's growing at 25 degrees Celsius, and then you get this sort of 4-degree warming, you can see that you've had a slight stimulation in photosynthetic rate. But the same sort of plant that's operating at 29 degrees Celsius to start with, and the same 4 degrees Celsius warming, you get a suppression of carbon uptake rates. And just like with CO2, the ability to model this and predict what plants are going to do in the future is made more complicated by the fact that photosynthesis acclimates to temperature. So if you have a plant that's grown at low temperature, that's what you see in this blue line here. Its photosynthetic rate is maximized at cold temperatures. And if you take the same plant and you transfer it to higher temperatures and let it develop there, then you'll see this shift along the x-axis where the temperature response curve is basically translated. That improves your performance at high temperatures, but of course it's at the cost of reducing your performance at low temperatures. So this is this classic view of what we expect to see with acclimation to high temperatures. And we've known about this for quite a while. This is a picture from a really classic paper from Berry and Bjorkman. It's got, I don't know, like 2,000 citations to it at this point. And this is some of the first data looking at this. These are desert plants that they got. This is the Carnegie Institute, so they're surrounded by deserts. And if we look at the data on the bottom, here we have photosynthesis compared to leaf temperature. And they've grown plants either at a cool temperature in black, and that cool temperature for them being desert is 22, or at a hot temperature, and we're talking here 42 degrees Celsius. Okay. In these three species at the bottom, you can see that very classic thermal acclimation response, where you just sort of see a translation along the x-axis of the curve. But you can already see in these very early data that there's a lot of diversity in that response. So in these two species, if you grow them at hot temperatures, photosynthesis collapses, and you never get enhanced performance at any leaf temperature compared to the cool grown plants. Similarly, if you have a sort of heat specialist like here, if you grow that at cool temperatures, then photosynthesis always does more poorly than in the warm grown plants. So it's not a general pattern. This is something where there's a lot of diversity. And we can characterize those curves looking at a few different points. So the first one is to look at what is the actual rate of photosynthesis at the growth temperature. And so in this case here, if you take the cool growth temperature in the cool plant, there's its photosynthetic rate compared to the warm grown plant measured at the warm temperatures. And that's what we refer to as a growth, assimilation at the growth condition. The other thing you can look at is the thermal optimum. So in other words, what's the temperature where this photosynthesis is maximized? And so for the cool plant, it's here. For the warm plant, it's here. And that's our T-opt. So we pulled together data from every study we could find that grew plants at different temperatures and measured these curves and looked for patterns. So across 130 different species, we found that the majority of them are able to maintain that photosynthetic rate at the new growth temperature, so that A growth parameter. And you can see that down here. If you look at all the data, then in 68% of the cases we looked at, so in 115 data sets out of 169, we saw this maintenance of the photosynthetic rate at a similar rate in the warm and the cool grown plants. But I want to emphasize that there's a fair bit of diversity, and I'm just going to emphasize the trees because that's what we're going to talk about later. Where deciduous trees did this almost all the time, although it's a fairly small data set. Whereas evergreen trees like boreal conifers only did this about half of the time. So in other words, their photosynthesis didn't acclimate as effectively as in deciduous trees. If we look at that thermal optimum characteristic, then again, you can see that the vast majority of the time, the thermal optimum shifts up with an increase in growth temperature. But again, there's diversity. And again, if we look at deciduous trees, they're very good at this. This is almost universal. But again, if we look at these evergreen trees, that only happens in about 2/3 of the studies that we looked at. And if you want to think about that classic thermal acclimation shift where you get a shift of the whole curve, then really what you need is for both of these to happen. You need to maintain that same rate on the y-axis and yet increase the thermal optimum. And when we look for that, which we refer to as sort of constructive acclimation, actually only half of the studies across the board, do you find that pattern? What we think of as being very classic and very common actually only happens half of the time. And again, deciduous trees, very small sample size, but they tend to acclimate quite well, whereas evergreen trees do not. So the point is that if we look at the optimum temperature for photosynthesis and the growth temperature, here we can just focus on these C3 plants in green. As you increase the growth temperature, you increase the thermal optimum. And you can see that in any data point here, where here we have a single study where we have the data from the cool growth temperature connected to the data from the warm growth temperature from that study. So anything with a positive slope means that you have that positive relationship. But thermal acclimation doesn't necessarily improve the actual amount of CO2 that a plant is taking up. And that's the thing that really matters when we're thinking about climate change and the ability to moderate climate change. And so here what we've done is we've got similarly growth temperature, and then we've relativized the photosynthetic rates, taken a value of 1 at the low growth temperature. And so anything with a positive slope means that photosynthesis increased at the warmer growth temperature. And any of the data points connected with a negative slope, you can see instead that photosynthesis was suppressed by warming. And especially for these C3 plants, that average is at around 1, but there's a lot of studies where you see a suppression of photosynthesis. So if photosynthesis doesn't maximize the amount of carbon that a plant is going to take up and doesn't maintain a high CO2 assimilation rate, what does it do? And some of the more recent work we've done shows that acclimated plants are actually just more likely to operate near whatever their maximum photosynthetic rate is. So that maximum photosynthetic rate may be suppressed by warming, but plants are more likely to operate near it. And you can see that here, where here I've got a figure showing the mean temperature that a leaf is experiencing and how much temperature variability there is. In other words, how fast does leaf temperature change? And the scale here is an index of how much time does the leaf spend at or above 80% of its maximum photosynthetic rate. So the short version of this is that yellow is good, it means the plant is doing well, and purple means that carbon assimilation is really suppressed. So for a non-acclimated leaf, you can see there's a fairly narrow window of leaf temperatures and leaf variability in temperature that promote high photosynthetic performance. But if you take an acclimated plant instead, what you see is a much broader suite of conditions where the plants are performing near whatever their optimum rate is. So if we translate those ideas about how warming influences photosynthesis, you might not be too surprised to hear that warming also differentially affects the growth of different types of trees. And that's shown here, where you can see here the percent change in either the shoot height, the stem diameter, or the total biomass of deciduous plants in yellow or evergreen woody plants in green. And the numbers at the bottom just show how many studies have gone into this sort of meta-analysis result. So this is what we would expect under that sort of global mean warming of 3.5 degrees Celsius. You might expect an increase in biomass in these deciduous species by perhaps 10%, but a decrease of about 22% for evergreen species. So we expect that under a future climate, deciduous trees may increase their growth, but in evergreen species, we may often see a suppression. So in sum on this part, looking at how will climate change affect plants, Higher CO2 is expected to stimulate photosynthesis and growth, but acclimation to higher CO2 reduces that benefit. And warming could increase photosynthesis and growth in cool regions, and especially in deciduous species. But acclimation to higher temperatures doesn't necessarily mean that you're actually going to have higher CO2 assimilation rates or higher growth. So what happens when we put these things together? And a lot of the work in our lab has focused on boreal forests and boreal species. And so this is just that distribution looking down at the top of the planet. And this is because they're a key player in the global carbon cycle. So about a third of the world's forest carbon stocks are locked up in this system. And there are relatively few tree species. There's low biodiversity. So the idea is that this is actually a tractable system where these dominant tree species have really large impacts on the carbon fluxes of the entire ecosystem. But there's so few of them that you could potentially study all of them and get a good handle on how that ecosystem may respond to climate change. So we focused a lot on two very common North American species. The first is black spruce, and you can see it in these pictures here. It's the one that's got the green foliage. And then tamarack is a deciduous conifer. They're not very common, but you can see it here in its fall foliage, where it's sort of these yellows and oranges colors. So this work has been done at the Biotron Center for Experimental Climate Change Research here at the Faculty of Science. And for those of you who haven't been here, here's a bit of a picture of it. We have these 6 rooftop glass houses with CO2 and temperature control, where we can grow plants under future climates and then look at how that affects their physiology and survival. And so what we've done is grown these plants under either current CO2 or elevated CO2, and then combine that with either current temperatures or a +4 or a +8 degree warming. So remember, this is the sort of temperatures we would actually expect by the middle of the century or the end of the century in these regions. So some of our most recent work has been done on these 2 species, and you can see here that in black spruce, warming suppresses leaf nitrogen concentrations. And so on the left-hand side, AC stands for ambient CO2, EC stands for elevated CO2, and then the control temperature, the cool is in blue, purple is your +4, and red is +8. So you get towards more red as you get warmer. You can see this suppression in leaf nitrogen. You see the same thing in tamarack. And so less nitrogen in these warm-grown trees should correlate with lower enzyme concentrations and therefore lower photosynthetic abilities, lower photosynthetic capacity. And that is indeed what we see. So this parameter here is VC max, is an index of the capacity of a leaf to fix CO2. How fast can it fix CO2 at ideal conditions? And again, if we take a look at black spruce and we measure this across a range of leaf temperatures, you can see that in the control plants, every time you increase, every time you increase the growth temperature by 4 degrees Celsius, you further suppress the ability of the plants to fix CO2. And you see this both under ambient and elevated CO2, and we see this both in black spruce and in tamarack. Lower leaf nitrogen concentrations, lower photosynthetic capacity, but much less of an effect of CO2 than of warming. So if you have less ability to fix CO2 inside the leaf, if that demand side of that equation is reduced in warming, then you should have lower photosynthetic rates. And that is indeed what we see with black spruce. So here's black spruce here. And again, the plant's grown at ambient CO2 or elevated CO2. So if we look at the photosynthetic rate across different leaf temperatures, then again, as you increase the growth temperature, the ability to fix CO2 is suppressed. And just like we talked about earlier, these plants grown at the higher temperatures never perform better at high temperatures, high leaf temperatures. So there's no trade-off here. It's simply that you get a general reduction in the performance of the plants. And we see something quite similar in the plants grown at elevated CO2. But in tamarack, you actually don't see that. So in tamarack here, plants grown at ambient CO2, it doesn't matter which leaf temperature they're grown at, they have very similar photosynthetic rates. And we see something similar at elevated CO2 where there's a suppression in the +4 grown plants, but then the plants grown at +8 degrees Celsius bounce right back up. So what we saw was that in both species, there's less leaf nitrogen and a reduced capacity to fix CO2. But photosynthesis is dictated by both that supply and that demand component. And so what that implies is that there's differences in the supply function. And that's actually what we see. So here is stomatal conductance, the ability to move CO2 into the leaf. And in black spruce from both ambient and elevated CO2, if you grow the plants at higher temperatures in the purple and red, you can see that they have lower stomatal conductance. In other words, there's a greater restriction for CO2 to move into the leaf. But in tamarack, what they're doing is they're offsetting the reduction in photosynthetic enzyme concentrations by increasing their supply of CO2 into those enzymes. So the plants grown at high temperatures actually have higher stomatal conductance than do the plants growing at the cooler temperatures. So one of the questions I get quite a bit is, I do a lot of these pot experiments in glass houses, and the question is, how realistic is that? If you go to Metro, you can probably buy one of these, but if you measure it, it doesn't necessarily tell you much about what's happening in a system like this. So how translatable are these results? So we're really quite fortunate that we're involved in a large-scale experiment in the US that's acronym is Spruce. And so Spruce stands for Spruce and Peatland Responses Under Changing Environments. And this is just a picture of it, an aerial view, where there's these 10 large open-top chambers surrounding sort of 40 to 50-year-old trees in an intact boreal forest. And this is what it looks like from the inside. So here are some of these trees. You can see the chamber around it. And this is one of the plots that they didn't put walls up around. And there are 5 temperature treatments in the experiment. So they range from 0 to plus 9 degrees Celsius. And there's now also a CO2 treatment on these trees, but that was not initiated at the time that we collected this data. So I'm going to show you some data from this site to make that correspond with the glasshouse work, because these are the same 2 species that we grew in the glasshouses, being grown under a similar range of temperatures. But here they're out in the field and they're large trees. So just to set the scene, we went out 3 times over the summer. And because the biological activity of the leaves shifts over the year, there's phenology, some of the data has been relativized within each month that we went out, just to look at the treatment effects and sort of emphasize those. And so you can see that here in the first set of data. This is the leaf nitrogen concentrations. Here we have the temperature treatments at the bottom. Tamaracks on the left, black spruces on the right. And the different shapes indicate the different months that we went out and measured the trees. So in tamarack, there were no leaves the first time we went out in the field because they're deciduous, they hadn't grown yet. Whereas with black spruce, we had 3 measurement campaigns. And the thing I think you can see quite clearly is that as you increase the temperature, these warmer plots over here, the leaves have more leaf nitrogen in them. And if you're still hanging on with me here, this is the exact opposite of what we find in the glasshouse. And I think this is one of the important sort of considerations when you're trying to scale this work about the difference between warming effects on plants versus those on ecosystems. This increase in leaf nitrogen concentrations in the warm plots is due to the fact that warming is stimulating microbial activity in the soils, and it's stimulating nitrogen mineralization rates. So it's sort of like you're combining a warming experiment with a fertilization treatment. So the plants in the warmest plots are getting not just an increase in temperature, but they're also getting an increase in nitrogen supply being put on top of that. So I think that the results from the glasshouse show that when you give all the plants the same amount of nitrogen, leaf nitrogen declines with warming. But that's basically being overridden by that stimulation of nitrogen in the soils out in the ecosystems. And the thing I do want to show you is that those stomatal responses between the species are very similar in these large-scale forests as they are with the glasshouse studies, though. So here we're looking at stomatal conductance, that ability to move CO2 into the leaf. And here we have data from black spruce measured across a range of leaf temperatures. So the warmest plots are shown in these darkest red colors. And you can see that as you get into these warmer, drier plots, the plants are closing their stomata, which restricts CO2 movement into the leaf. They're doing this, of course, to actually prevent excessive water loss. But the other effect of that is that it restricts CO2 diffusion. So because these warm-grown plots also have higher nitrogen, they have a high demand for CO2 inside the leaf, but they're restricting that supply. And the net effect of those 2 things combined against each other is actually a remarkable consistency of photosynthesis across this 9 degrees Celsius growth temperature. But we can use an index of stomatal limitations of photosynthesis, and that's what's shown down here. This is intercellular CO2, how much CO2 is inside the leaf. And if that declines, it indicates that stomata are playing a key role in restricting the rate of photosynthesis in these plants. So as you get into these warmer plots, what you see is a greater control of stomata on photosynthesis compared to the demand function. Now for tamarack, what we see is actually the opposite. And in fact, this is the only species I've ever seen this in, where if you grow plants at higher temperatures, even though it's also drier, they open their stomata more. And so the plants in the warmest plots have more leaf nitrogen, more photosynthetic enzymes, and also a greater supply of CO2. And the net effect of both of those is that you see a stimulation of the photosynthetic rates and a balance between supply and demand that leads to a sort of a flat response of that CO2 concentration inside the leaf. So tamarack is keeping its stomata open, which is reducing those stomata limitations for photosynthesis. It's prioritizing carbon over water. But spruce are closing their stomata under warming. It's limiting photosynthesis, but it's maintaining their hydraulic balance and preventing excessive water loss. So across both of these experiments, we're seeing that the stomatal responses to warming between these two key species differ, but they differ in the same way, right? Tamarack are opening their stomata and spruce are closing them. And so in both experiments, photosynthesis is less negatively affected by warming in tamarack than it is in the spruce species. And more recent data that we have from the site also shows that while we get these strong warming effects, that the CO2 effects tend to be much weaker than are the effects of the elevated temperature across this gradient. So I've talked a lot about photosynthesis, and I just want to briefly say that we're finding this sort of response where temperature is predominant over CO2 in quite a lot of different systems and in different processes that we look at. So just as a one-off, this is a measure of respiration, so CO2 efflux from a leaf across a range of temperatures. And again, these are plants grown at either ambient CO2 or elevated CO2 in the biotron, grown at either current temperatures, +4 or +8. And you can really clearly see, of course, that as you increase the growth temperature, you get this suppression in respiration, but that the effective CO2 is much weaker. And more recently, we've expanded the number of species that we're looking at. And I could show you more graphs, but I think it's nice to see some pictures of these things. And so we have a suite of different boreal species that we're looking at. And what you have on the left-hand side are all the plants grown at ambient CO2. And then you have the current temperature, a +4 and a +8 warming. And then on the right-hand side, you have EC, the elevated CO2 plants, current temperatures, +4, +8. And in all of these species that we're looking at, whether it's the deciduous conifer, the evergreen conifers, or the deciduous broadleaf species, the +8 degrees Celsius warming suppresses growth. Growth is generally enhanced by elevated CO2, but it's really the deciduous species that are doing the best under those sort of future climate conditions. You can look down here at paper birch and see that at a +8 degree warming, growth is not too much suppressed compared to current conditions, which are here. But in other species like Picea mariana, black spruce, or white spruce, or here in jack pine, you can barely see the seedlings compared to the pot, compared to what they are in the current conditions. And we're also seeing a lot of chlorosis, browning of the plants. This is a plant growing at +8 with elevated CO2. This is a jack pine. They're fully watered, they're fully fertilized. So really this is a direct effect of seeing these extreme temperatures compared to things like drought, which we actually expect in the field. And unsurprisingly, when you see plants that look like this, we get high mortality rates. So I just want to conclude by saying that elevated CO2 may not lead to these widespread productivity increases, especially when combined with warming, that many people might expect. And moderate warming can boost growth and carbon uptake, especially in deciduous species, but that's much less of a potential for evergreen conifers, which dominate these boreal forests. And the warming predicted for the end of this century is so extreme, it's likely to suppress growth, even with high CO2 and even in these deciduous species. And this is just a figure showing the sort of climate change we've actually already had since about 1950 in Canada, where you can see at these higher latitudes, we've actually already gotten 3.5 degrees Celsius of warming. So this isn't something that we're expecting in the future, it's something that's here now. So with that, I'd like to thank you guys for listening and showing up. I realize it's the middle of the semester and everybody's really, really busy. So thank you for coming. And also all the people who've helped with the work and also helped by supplying funding for us. Thank you.
Thank you so much, Danny, for a really important and informative talk. I see lots of hands clapping. So are there any questions for Danny? If you could please raise your hand and I will try and keep an eye on the participant list. And we'll take questions. I meant raise your Zoom hand, not your actual hand, because I can't see everyone on my screen. No questions yet?
I have a question.
Go ahead. That's Keith. Go ahead.
Yeah, hi, Danny. Thanks. Great talk. I may have mentioned this before. We discussed this with you before, but I'm curious about the interaction between water use efficiency and CO2 uptake in leaves. And it seems like we're modeling temperature only and CO2 availability. And I wonder if people are going in the direction of thinking about relative humidity and water stress of plants. You can have hot, dry, and hot, wet. And so, how do those scenarios play out in your modeling?
Yeah, so those would be very different capacities for the plant in the sense that obviously if you have a hot, dry condition, in some species, the stomata almost completely close, or they do completely close to prevent catastrophic failure of the water transport system, of course. Whereas if you have considerable amounts of soil moisture, you may be able to maintain them open, right? The thing that we're seeing, and we actually just got this paper accepted this week, so I was quite pleased with it. We just recently looked at some data where you've got well-watered plants, and instead what you're looking at is water stress being induced by high vapor pressure deficit, so atmospheric drought, basically. The plants have lots of water around their roots, but the air is very dry. And we see a lot of these same suppressions of growth that you see in drought experiments. And so it's quite interesting that even though they have water with their roots, they may not be able to get it to their leaves with sufficient efficiency to be able to maintain a well-hydrated leaf and therefore maintain physiological function. So you get suppressions in, for example, plant height, plant biomass, yield in crops. So there's quite a broad range of effects from that, just that alone.
So just briefly, what do we know about the climate change predictions in terms of hydrology and ambient water versus root water, as you've just described? I mean, is that a complete unknown, or are there people modeling that?
Yeah, so there's a lot of work on modeling that. It's not so much what I do, but the precipitation predictions for the future are always a little bit more uncertain than are the temperature predictions. There's a lot of convergence in the temperature predictions in these models. The general expectation, to my knowledge, is that relative humidity will stay the same in the future climates. And what that means is that as the temperature increases, if relative humidity stays the same, then that vapor pressure deficit actually becomes greater. And so atmospheric demand for water from plants will increase under future climates. That is one of the predictions.
So it seems that not everyone is able to raise their hand. Maybe nobody is. Oh, Chris and Yolanda, some people can raise their hands. If you aren't able to raise your hand, then I suggest you just yell out your question, but Chris or Yolanda has a question right now. Go ahead.
Hey, Danny.
Hey, Chris.
Thanks, that was really nice. When you talk about the range shifts that are expected for these species, so deciduous species moving north and range contractions in coniferous species, and those are supposed to happen over such short time frames, like 50 years compared to the lifespan of these trees. What does the future look like if the coniferous species can't survive, but the deciduous species can't even move north fast enough to fill in? Like, will they just replace? Or will the northern forests become a bunch of saplings and dead pine spruce trees?
Yeah, I think when you get to those sort of ecosystem questions, there's a lot of other things that feed in. I mean, there's increases in the fire cycle because of the increasing temperatures and the dryness in these latitudes, and increased insect outbreaks as well. If you're just thinking about the ability of the species to move, those models I showed you at the beginning of the talk are what we refer to as climate envelope models. So they basically say, here's where you find a plant growing now, and here's the sort of temperature and precipitation conditions it prefers, and here's where that temperature and precipitation will be in the future. But of course, that doesn't necessarily mean the species will have the correct soils. It doesn't mean it will have the ability to migrate to get there. So those are sort of almost more like best case scenarios of what you'd expect if the species can move and it can find the appropriate conditions other than just temperature and precipitation. So there is a lot of question about how those dynamics will play out. And we are seeing sort of an increased shrubification at high latitudes, where areas that used to be more dominated by, for example, grasses or evergreen species, you end up with this sort of like a lot of the shrubby deciduous species moving in. They tend to establish faster and they grow better. And so there have been a number of recent remote sensing sort of studies that have shown increased greening in, at really high latitudes, but browning in the mid-boreal forests. So the idea is that basically this sort of increasing drought stress and heat stress is suppressing the growth and creating mortality for these boreal conifers. But in some places that's being offset by increased growth by things like, birch and poplar and some of the willow species. Right. So I expect there'd be a lot of change in sort of what the structure and the function of that system is going to look like. And we know that if you go back in time, you've had completely non-analog environments, right? Like Fred told me he couldn't make it today, but like Beringia, where you had these sort of tundra steppe-like ecosystems that we just don't have an equivalent for now. So it's such an extreme change that I think we don't really know what that's going to look like.
Right. Thank you.
Lars, go ahead.
Yeah, Danny, great talk. I'm curious, how does the scale of agricultural carbon fixation compare to this natural forest and ocean-based carbon fixation? And related question, would it be possible, you think, to use agriculture as carbon capture technology and just grow something, store it in the desert for 1,000 years, maybe?
Yeah, so that estimate of terrestrial photosynthesis is really all terrestrial systems. So agriculture is a component of that number already. There is a lot of talk about sort of carbon capture, of course, and the idea of being able to, use species that have high photosynthetic rates and put that carbon into sort of more recalcitrant tissues that don't break down as easily, and store that in things like biochar or in other sorts of systems. And you may have heard of that sort of 1 billion tree initiative. That was the idea of if we could plant 1 billion trees, that would absorb a lot of the CO2. And at least over shorter timescales would give us a little bit of breathing room in trying to meet that transition to a green energy economy. So over shorter timescales, this sort of thing definitely works. The problem is, of course, if you grow a whole new forest and you're using that as your main way of reducing CO2 emissions, by instead increasing CO2 absorption, all it takes is an insect outbreak or a fire or a drought, and that whole thing could end up right back into the atmosphere due to respiration, right, in short timescales. So I think those sorts of things are useful tools, especially over the next 50 years while we're doing this transition, but I don't think they can be the solution, so to speak. They all play important roles, and there's a lot of emphasis on these sort of more nature-based, natural-based climate solutions. And so again, they can help us get there, but the main thing really is that transition that we have to make.
Thank you.
Any other questions? Robert?
Hi, thanks very much for the talk. I'm interested, I've heard some talk in the community land model community about building a new version of the community land model that sort of tracks plant functional types in a lot more of an explicit way, right? This is the FATES idea that they've been working on. Are you optimistic that that will be able to capture some of these differences in photosynthesis and respiration responses? Is that the direction we should be going in, or is that going to be just too complicated to parameterize and be confident that we're getting the biology right?
Yeah, so just to pull that out for people who aren't in the field, the idea is that these large scale models, like the Earth system models, simplify the biology of the ecosystems by using a plant functional type approach. So the idea is that a deciduous tree is different than an evergreen tree, which is different than a grass, for example. And then they have different types of subdivisions of how fine they get with those divisions. So yeah, as you add more plant functional types, of course, you have to actually have enough data to parameterize their behavior under all these different scenarios. So I know there are some moves to that. And that's actually part of where that study that I showed at the end with the multiple species is going, is we're trying to characterize within a plant functional type, for example, how much diversity is there in the response to CO2 and temperature? And is that important to be able to catch in these models? I guess my thing is that I realize that the Earth system models have so much complexity in them already that every time you add more, the computing power alone is just enormous. I kind of like the move towards things like Mike Dietz's work, where you have instead probability distribution functions for these plant functional types. So instead of having a single value for each parameter for a wide range of species, that instead you recognize that there's diversity even within any plant functional type, and you build it in through that sort of function, rather than constantly continuing to split into smaller silos that each have their own parameterization, which would have such huge error bars around it anyways, that we're maybe not improving the accuracy of the model. But yeah.
Very cool, thank you.
Any other questions? So I have a question for you, Danny. I was just wondering, how important do you think in the long term the sort of change in like mean temperatures is going to be compared to potentially these just spikes in like more extremely high temperatures, for example, that might happen once in a while? How much do those extreme events affect plants?
Yeah, I think the extremes are actually probably more important on an ecological scale, because, you know, you get an extreme heat and drought and you have mass mortality of plants, and you get a complete transition to a new type of system. That's a much bigger effect on the landscape level than will be the fact that growth is suppressed 20% on average, you know, over years, and it accumulates. And those sort of large scale extreme events are of course happening more often too, whether it's extreme heats, extreme droughts, again, insect outbreaks, fire cycling, all of those sort of things play into it. So again, this is one of the other things I sometimes get feedback on is, well, what does it matter if the plants grow, if they're less productive when you grow them at +4, since it doesn't really matter. It's really gonna be the things like the fires and such that dominate that signal at the ecosystem level. But of course, not everything is disturbed every year, right? And so that patchwork mosaic starts to come out at that global scale, where it's really important to capture both of these effects, I think. You know, these wide scale, those stochastic events are going to be really important, but so is the general productivity of all the other vegetation that's not undergoing that disturbance. So, but yeah, they're both big players.
Thanks. Any other questions for Danny? Nope. Anyone want to shout out if they can't put their hand up? Now's your chance. Okay.
It
well,
is
see.
People
Sorry?
are worried about the next meeting. It's 2:30. People are worried about the next meeting.
Yeah, I guess. I thought this was scheduled till 3 though. I thought we had more time.
Yeah, I think the meeting sort of is, but yeah.
Anyway, thank you so much, Danny, for a really, really interesting and accessible lecture. Please join me, everyone, in thanking Danny and congratulating her again on receiving the Florence Bucke 2020 prize.
Thanks again. I really appreciate everyone coming out. So thank you very much.
Thank you and congratulations, Danny.
Thanks.
And thank you, everyone, for coming today.
And thanks, Ganusha, for such a lovely introduction. Thank you.
I could have gone on longer, but they're not here to hear me. Great job.